Welcome to the Deep Dive. Today we are peering into the very fabric of existence, investigating really the most profound process on Earth, evolution.
Right. It's the story of how life adapts, changes, and transforms. It's given us everything from the simplest bacteria to, the complexity of the human brain.
And when you stop to think about it, life presents this incredible paradox, doesn't it?
It really does. You can marvel at the staggering diversity. I mean, tens of thousands of beetle species, or all the bizarre adaptations of deep sea fish, or the perfect camouflage of an orchid mantis.
Just incredible variation.
But beneath that boundless variation lies this deep, undeniable unity. All mantises, for example, share that fundamental six-legged structure. All life shares the same genetic code.
That tension between the infinite variation and the underlying unity is exactly what we're tackling today. Our mission is to take a deep dive into the mechanisms of evolution. We're going to try and uncover the foundational patterns that shape life, the processes that drive genetic change, and the 4 billion year history written in the rocks and in our own DNA.
And I think a great place to start is with the highest level question, which falls under the umbrella of evolutionary developmental biology, or Evo-Devo.
Okay.
We want to understand how simple, single-celled ancestors could possibly give rise to this massive, complex, multicellular life we see all around us.
And Dr. Cassandra Extevor's work on this topic, it raises the central mystery brilliantly. If you take any multicellular organism, a fly, a coconut tree, or yourself, every single cell contains the exact same set of DNA instructions.
Same complete genome, right?
So if the DNA blueprint is identical, why are some cells in your body performing, complex rapid signaling as nerve cells?
While others are synthesizing enzymes as liver cells,
Yeah.
and yet others are forming physical barriers as skin cells.
Exactly. It's the problem of differential gene expression.
It is. And when you scale that up to evolution, the question becomes, how have changes in that gene function over time created the immense morphological differences we see today?
So how do you get from, say, a coconut to a rose?
Precisely. They both share this ancient machinery for sexual reproduction, but the final forms were just worlds apart. Or think about a monkey embryo versus a human embryo. For most of the early stages, they look nearly identical.
Almost indistinguishable.
But the final adult forms are so vastly different. So the key isn't necessarily inventing entirely new genes.
It's about changing the orchestration, the timing, the regulation of the existing genetic toolkit.
That is the core insight of Evo-Devo. Extevor's research on germ cells, the eggs and sperm, illustrates this perfectly. They found that in many diverse species, the same set of ancient homologous genes are used to generate these crucial germ cells.
So a conserved function.
In that case, yes. But then they looked at other cell types and found cases where closely related organisms co-opted those same homologous genes for entirely new non-germ cell functions. Evolution is the ultimate opportunist. It's, reusing and modifying existing instructions rather than constantly writing new code from scratch.
Okay, so that sets the stage beautifully for the big history. To really appreciate Darwin's insight, we have to understand just how radical the idea of change was back in the 18th century.
It was completely revolutionary because the intellectual landscape was just dominated by fixity. Think back to Aristotle's scala naturae, the scale of nature.
The great chain of being.
Right. It placed all life on this fixed, unchanging ladder of complexity, with humans at the top, of course. This concept suggested every species was perfect, permanent, and had its predetermined place.
And even Carolus Linnaeus, the guy who gave us our classification, system, genus, family, order.
He used it not as a reflection of evolutionary kinship, but purely as a system to categorize the perfection of creation.
But the fossil evidence started to poke holes in this fixed view.
It did. Georges Cuvier, studying the rock layers, the strata, noticed these distinct boundaries where many species just disappeared and new ones appeared. He was a brilliant anatomist, but he used these gaps to support catastrophism.
He argued that global catastrophes wiped everything out.
Exactly. And then the area was just repopulated by species immigrating from elsewhere, so he was actively denying gradual evolutionary change.
Then you have Lamarck, who actually got halfway there. He recognized that evolution occurs and that it explains the adaptations we see, like the long neck of the giraffe.
Right, but his proposed mechanism was fundamentally flawed.
Use and disuse.
Lamarck proposed use and disuse. If you use a trait a lot, it gets stronger. If you don't, it shrinks. And critically, the inheritance of acquired characteristics. The idea that traits you pick up during your lifetime could be passed to your kids.
And the classic counterexample is the bonsai tree.
The perfect one. You can prune and shape that tree for generations, but the seeds it produces will still grow into a normal full-size tree because those physical changes don't affect the DNA in the seeds.
So Lamarck saw the pattern, but got the mechanism wrong. Darwin's breakthrough was figuring out that mechanism. How did his famous journey set the stage for that?
Darwin's 5-year voyage on the HMS Beagle was the catalyst. He brought along Charles Lyell's Principles of Geology, which argued for uniformitarianism.
The idea that geological processes we see today have been happening at the same rate for a very, very long time.
Exactly, which implied the Earth was enormously old. And Darwin saw this firsthand when he witnessed a violent earthquake in Chile. He saw the coastline physically uplifted, and later saw marine fossils high up in the Andes Mountains.
That must have been a profoundly humbling moment, seeing immense geological change happen instantly.
It really was. It confirmed that tiny, gradual changes over vast periods of time could shape the entire planet. And this deep time context allowed him to view biological change through that same lens.
So he started focusing on adaptation.
All of his observations focused on adaptation-inherited traits that enhance survival and reproduction in a specific environment. The unique beaks of the finches on the Galapagos, perfectly tailored to the local seed types, showed him that adaptation and the origin of new species were inextricably linked processes driven by natural selection.
And as we know, he wasn't the only one thinking this way at the time.
No. Alfred Russel Wallace, who was studying in Southeast Asia, independently arrived at the exact same concept. He sent Darwin his manuscript in 1858, and that really spurred Darwin to synthesize his decades of research and publish on the origin of species.
So let's nail down the core logic of natural selection. It's so elegant, really. It starts with two simple observations.
Observation 1 is variation. Members of a population vary in their inherited traits. This is the raw fuel. If you look at Asian ladybird beetles, some are red with 2 spots, others are orange with 6, some are black with none. They're all slightly different.
And observation 2 is overproduction and competition.
Yes, this was inspired by Thomas Malthus's work. Darwin realized that species produce way more offspring than the environment can possibly sustain. This inevitably leads to a struggle for existence, where many offspring just fail to survive or reproduce.
Okay, so you combine inherited variation with competition. That gets you to the two crucial inferences.
Inference 1 is differential survival and reproduction. Individuals whose inherited traits give them a survival advantage or maybe make them better at finding mates will, on average, leave more offspring than others.
And the second inference follows directly from that.
It does. Accumulation of favorable traits. This unequal success in reproduction leads to those advantageous traits accumulating in the population across generations. And that is the very definition of adaptive evolution.
This model immediately clears up two really common misunderstandings about evolution. First, selection doesn't create new traits.
It's an editing process. It's so important to remember that. Natural selection can only act on variation that's already present in the gene pool. It can't just conjure up a new defense mechanism because an organism needs it.
And second, individuals don't evolve.
The entire population evolves as the frequency of alleles changes over time. You don't evolve in your lifetime. The population your descendants belong to will be different.
Okay, so we've covered the theory. Now for the evidence. We rely on 4 fundamental lines of evidence to confirm the pattern of evolution. Let's start with the most dramatic, direct observation of it happening in real time.
The soapberry bug studies are a fantastic example of rapid directional selection. These insects feed on seeds inside host plants using a needle-like beak. In southern Florida, they fed on the native balloon vine, which has these large fruits that require a long beak to pierce.
But then a new plant was introduced in central Florida.
Exactly. The golden rain tree, a non-native species, which produces much smaller, shallower fruit. Researchers watched as soapberry bugs colonized this new food source. And over just 35 years, a tiny span in evolutionary terms, the population feeding on the golden rain tree evolved significantly shorter beaks.
Because shorter beaks were more efficient for that new food source.
They were. It's a perfect, documented, measurable example of a population adapting to a new environment.
And probably the most urgent example for us today is the rapid evolution of drug resistance in bacteria.
Oh, absolutely. Consider Staphylococcus aureus. When penicillin was introduced broadly in 1943, it was a miracle cure, but resistance appeared within 2 years. Why?
Because some bacteria already had the resistance gene.
Some S. aureus individuals already possessed a gene coding for penicillinase, an enzyme that just destroys penicillin. The drug didn't create the enzyme, it selected for the few bacteria that already had it, allowing them to proliferate.
So when methicillin was introduced to fight that,
Same story, even faster. Methicillin was designed to resist penicillinase, but again within 2 years we saw methicillin-resistant S. aureus, or MRSA. This strain had evolved a different mechanism, an enzyme that let it build its cell walls using an alternative pathway, one that methicillin couldn't block.
it's a constant evolutionary arms race.
It is. The antibiotic is just a powerful selective agent, favoring the survival of the already resistant individuals.
Okay, our second line of evidence is homology similarity that results from shared ancestry. This is where comparative anatomy comes in.
And the classic illustration is the mammalian forelimb. If you compare the skeleton of a human arm, a cat leg, a whale flipper, and a bat wing,
They're all used for totally different things.
radically different functions, lifting, running, swimming, flying, yet they all share the exact same underlying bone structure, one upper arm bone, the humerus, followed by 2 forearm bones, the radius and ulna, then wrist bones and finger bones.
These are homologous structures.
They're variations on a common structural theme that was present in their shared tetrapod ancestor.
And we also see these evolutionary ghosts in vestigial structures.
Yes, these are remnants of features that served an important function in an ancestor but are now reduced and largely useless. Think of the tiny pelvic bones found deep inside some snakes, or the non-functional eye remnants buried beneath the scales of blind cave fish.
They make no sense in the modern animal.
None at all, but they're powerful evidence of descent from ancestors that did use those legs and eyes.
And this homology, it goes all the way down to the molecular level.
But the unity of life is absolute here. All known life forms, from viruses to us, use the same universal genetic code. We share homologous genes with bacteria. Some genes, like those for ribosomal subunits, have kept their function for billions of years.
And we even have vestigial genes, right? Pseudogenes.
Exactly. Inactive pseudogenes are non-functional genetic remnants inherited from ancestors, the molecular equivalent of a snake's tiny pelvis.
So when we combine all these homologies, we can use tree thinking.
Tree thinking reveals a perfect nested hierarchy. For instance, all organisms share the genetic code. All vertebrates share a backbone, but only tetrapods share that homologous limb structure. This pattern of increasingly restricted shared traits perfectly reflects common ancestry and divergence over time.
But we have to be careful. Similarity doesn't always mean shared ancestry. We have to address convergent evolution.
That's the vital distinction between homology and analogy. Analogy is similarity that evolves independently in different lineages because of similar environmental pressures.
Like the flying squirrel and the sugar glider.
The perfect example. One is a placental mammal from North America. The other is a marsupial from Australia. They look strikingly similar because they both adapted to gliding in forests at night, but their underlying anatomy and reproductive systems are vastly different. Their similarity is analogous, not homologous.
Okay, our third pillar of evidence, the fossil record.
The historical documentation of life. Now, the record is imperfect. It's biased toward common species with hard shells or bones, but what we have is invaluable.
And we can date these fossils pretty accurately now.
We can. We determine their absolute age using radiometric dating, which measures the decay rate, or half-life, of radioactive isotopes within the rock layers.
And the fossil record beautifully documents major transitions. The origin of mammals is one of the most compelling narratives.
It's truly stunning. The transition from synapsid reptiles, like the cynodonts, to modern mammals took over 120 million years. And the fossils clearly show the gradual evolution of key mammalian traits, like the shift toward a single lower jawbone and specialized teeth.
But the part that always blows my mind is what happened to the old jaw hinge bones.
Yes. In the cynodont ancestors, the jaw hinged between 2 small bones, the articular and the quadrate. As the main jawbone, the dentary, grew larger, those 2 ancestral bones shrank and were gradually co-opted for an entirely new function.
They moved into the middle ear.
They migrated into the middle ear, becoming the hammer and the anvil, working with the stirrup to improve hearing.
That's the ultimate example of evolutionary tinkering. It didn't design the ear from scratch. It repurposed a few bones it had lying around.
We see similar transitional series for whales, too. Fossils like Pakicetus documented the progression from four-legged, land-dwelling mammals to fully aquatic life, showing the gradual shortening of hind limbs and changes to the spine for swimming.
Okay, our final line of evidence, biogeography, the geographic distribution of species. This is a story written by geology.
It is entirely governed by plate tectonics, the slow movement of continental plates. About 250 million years ago, all the land was fused into the supercontinent Pangaea.
And when Pangaea broke apart.
It physically isolated lineages, setting up grand, global-scale allopatric speciation. This explains why certain groups, like the marsupials, achieved such unique dominance in geographically isolated places like Australia.
And this gives evolutionary theory predictive power.
It does. If we know that modern horses, the genus Equus, originated in North America about 5 million years ago, and we know from geology that North and South America weren't connected then, the theory predicts we should find older Equus fossils only in North America. And these kinds of predictions are constantly being tested and confirmed.
All right, we've established the pattern. Now we have to drill down into the engine room, the processes that change the genetics of a population, which we call microevolution.
And microevolution is defined simply as a change in allele frequencies in a population over generations. We have to remember the population is the smallest unit that can evolve.
So the prerequisite for any of this is genetic variation. Where does this raw material come from?
The fundamental source of new alleles is mutation, a change in the nucleotide sequence of DNA. Now, this can be a point mutation, like the one that causes sickle cell disease, which can be harmful, but often mutations in non-coding DNA or silent mutations result in neutral variation differences that provide no immediate advantage or disadvantage.
And the speed of mutation must be hugely important, especially in things like viruses.
Oh, absolutely crucial. Organisms with very short generation times and poor DNA proofreading, like the HIV virus, generate new mutations incredibly fast. That rapid mutation rate is why we have to constantly develop new drug cocktails. The virus is evolving resistance literally while we're treating the patient.
Sexual reproduction, though, doesn't create new alleles, but it's still essential.
It's the master shuffler. It creates unique combinations of existing alleles through three main processes: crossing over during meiosis, the independent assortment of chromosomes, and the random nature of fertilization. These processes ensure that every offspring is genetically unique, providing that wide range of phenotypes for natural selection to act upon.
So to measure if evolution is happening, we need a baseline, a null hypothesis. That's where the Hardy-Weinberg principle comes in.
Exactly. Hardy-Weinberg describes the genetic makeup of a population that is not evolving. We start by defining the gene pool as all the copies of every allele in that population. If a gene has two alleles, we label their frequencies P and Q. By definition, their frequencies have to add up to 1 P plus Q equals never 1.
So if we know the allele frequencies, we can use the equation to predict the genotype frequencies in the next generation, assuming nothing is changing.
The famous equation is 2p$ plus 2pq plus q2 equals 2. This predicts the expected frequency of the homozygous dominant individuals, Q22s, the homozygous recessive individuals, 2p$, and the heterozygotes, 2p$. If we measure the actual genotype frequencies in a real population and they don't match this, we have mathematical proof that evolution is happening.
And for this ideal, non-evolving state to exist, 5 very stringent conditions must be met.
1. No mutation. 2. Random mating. 3. No natural selection. 4. An extremely large population size to avoid random chance events, which we call genetic drift. And 5. No gene flow, so no migration.
Since real populations always violate at least one of these, Hardy-Weinberg is a model, but a very useful one, especially in medicine.
Oh, for sure. Take the metabolic disorder PKU. It's caused by a rare recessive allele, and its incidence is about 1 in 10,000 babies in the U.S. Since affected individuals are homozygous recessive, we set their frequency equal to $2.08.
So $2 is $0.0001,
Okay.
which means the allele frequency is the square root of that, or $0.01.
Exactly. And since P plus Q equals $1, the dominant allele frequency P must be $999. We can then estimate the frequency of healthy carriers, the heterozygotes, which is 2 picowu.
Which comes out to about 2%.
Nearly 2% of the population are carriers, silently concealing that harmful allele. Hardy-Weinberg is a powerful tool for estimating that.
Okay, we know selection is one of the 3 forces driving microevolution.
Yep.
Let's look at the second. Genetic drift. This is evolution driven purely by chance.
Genetic drift describes these unpredictable fluctuations in allele frequencies from one generation to the next. And the effects are massive only in small populations. If a population has millions of individuals, A few random deaths won't matter. But if it has only 50 individuals, the random death of 3 who happen to carry a rare allele can radically alter the gene pool.
And drift can have really devastating
It
consequences.
can lead to the random loss of genetic variation, or conversely, the random fixation of an allele, even a slightly harmful one, meaning it reaches 100% frequency just by chance, not because it was better.
So how does this happen in nature?
2 main phenomena, the founder effect and the bottleneck effect. The founder effect is when a small group gets isolated and starts a new population. By chance, their gene pool might not represent the source population at all.
And the bottleneck effect is a sudden, often catastrophic reduction in population size.
Think of it like pouring marbles out of a bottle. The few that get through, the survivors, may not reflect the original diversity. The greater prairie chicken in Illinois is a profound, real-world example of the damage a bottleneck can cause.
Tell us about that. What did researchers see after the population crash?
Well, due to habitat loss, the Illinois population of prairie chickens fell from hundreds of thousands to fewer than 50 birds by the 1990s. This severe bottleneck just wiped out their genetic variation. Critically, it also led to a huge decrease in the egg hatching rate, which fell to below 50%.
What did that signal?
It suggested that genetic drift had, by chance, increased the frequency of harmful fixed alleles that caused developmental failure because there just weren't enough healthy alleles left to mask them.
So how did they fix it?
The only way was to intervene using the third mechanism, gene flow. Researchers introduced hundreds of birds from healthy, diverse populations in neighboring states, effectively injecting new alleles back into the gene pool. And it worked. The egg hatching rate shot back up to over 90%.
Which brings us perfectly to gene flow, the transfer of alleles between populations, usually through migration.
And gene flow, unlike drift, tends to be homogenizing. Its usual effect is to reduce the genetic differences between populations, potentially welding them into one large combined gene pool.
But it can also work as a constraint, right? It can make it harder for a population to perfectly adapt to its specific environment.
The Lake Erie water snake is the perfect case study. On the mainland, banded coloration is great camouflage in marshes, so selection favors banded snakes. But on the rocky, open shores of the islands, selection favors unbanded, plain snakes.
But snakes swim over from the mainland.
Exactly. Every year, a few banded snakes migrate from the mainland to the islands. This gene flow continuously reintroduces the banded alleles into the island population.
So the island snakes are constantly evolving toward being unbanded, but the migration keeps pulling them back towards being banded?
Precisely. The flow of genes from the mainland prevents the island population from achieving perfect adaptation to its rocky habitat. It's a powerful example of how selection, drift, and flow are constantly in a dynamic struggle.
Okay, we've saved the deep exploration of natural selection for this section because it's the only one of the 3 mechanisms that consistently leads to adaptive evolution. It's not random.
Right, it's not random like drift, and it's not homogenizing like flow. It targets improvement. And we have to start by defining fitness rigorously. We must discard the idea of survival of the fittest as just being about strength or battle.
It's all about reproduction.
It's all about relative fitness, the contribution an individual makes to the gene pool of the next generation, relative to the contributions of others. A visually dull organism that lays 10,000 eggs has much higher relative fitness than a spectacular one that only leaves 3 offspring.
And selection acts on the phenotype, the organism's traits.
Directly on the phenotype and only indirectly on the genotype. And we can see how selection alters the phenotypic distribution of a trait in 3 distinct ways.
The first is directional selection.
This mode favors individuals at one extreme end of the range, pushing the entire population distribution toward that extreme over time. The finches evolving larger, harder beaks during a drought is the classic example of directional selection.
Next, we have disruptive selection.
Disruptive selection is fascinating because it favors both extreme phenotypes while selecting against the intermediate ones. Imagine a bird that eats seeds. If there are only very small seeds and very large seeds available, birds with intermediate beaks are inefficient at both. So selection favors both the small-beaked and large-beaked birds.
And finally, stabilizing selection.
This mode acts against both extreme phenotypes, squeezing the population around the intermediate mean. It tends to reduce variation. Human birth weight is the best studied example. Mortality rates historically increase for babies born significantly below or above the average weight of 3 to 4 kilograms. Selection favors that intermediate weight.
Okay, moving past basic survival, selection operates heavily on traits that enhance mating success, which we call sexual selection. This often results in sexual dimorphism.
Where males and females look drastically different. We categorize it based on who is competing and who is choosing. Intersexual selection is competition among one sex, usually males, for access to mates. This often involves fighting, displays of dominance, or guarding territory.
And intersexual selection is mate choice.
Right, where one sex, typically females, chooses mates based on the other sex's traits or behavior. This leads to the paradox of the peacock's tail, a massive, costly, dangerous trait. Why select for something that actively decreases survival?
Because the reproductive benefit outweighs the predation risk.
Exactly. The good genes hypothesis suggests females prefer traits that honestly signal high overall genetic quality.
And this was tested in the wild, right?
A beautiful experiment was done with gray tree frogs. Female tree frogs prefer males that perform long, loud mating calls. Researchers compared the offspring of long-calling males to the half-siblings produced by short-calling males, and they found that the offspring of the long-calling males showed superior survival rates and faster growth.
So the long call was an honest indicator of good genes.
It was. It confirmed the showy display was benefiting the female's offspring.
Natural selection doesn't always reduce variation, though. Sometimes it acts to maintain it, which we call balancing selection.
Balancing selection keeps multiple alleles or phenotypes present in a population. The first way this happens is frequency-dependent selection, where the fitness of a phenotype depends on how common it is.
And the African scale-eating fish is the perfect, strange example of this.
This cichlid fish feeds by sneaking up on other fish and ripping scales off their flank. Some have mouths twisted to the left, allowing them to attack the victim's right side, and vice versa. Prey fish learn to guard against whichever mouth type is currently most common.
So it pays to be rare.
It does. Selection favors the least common mouth type because it's the one the prey aren't watching for. This process keeps both the left and right mouth phenotypes at roughly 50% frequency.
The second mechanism of balancing selection is probably the most famous: heterozygote advantage.
If the heterozygote genotype has higher relative fitness than both of the homozygotes, then both alleles will be maintained in the population, even if one is lethal when homozygous. The classic example is the sickle cell allele in regions where malaria is endemic.
Right. Homozygotes for the normal allele get severe malaria. Homozygotes for the sickle cell allele suffer from the disease.
But the heterozygotes are spared from the worst symptoms of malaria, and they don't suffer the full effects of sickle cell disease. They have the highest relative fitness, which protects the population from both extremes and ensures that the potentially lethal sickle cell allele persists.
Okay, given the power of selection, it raises the ultimate question. Why doesn't evolution produce perfect organisms? Why do we still have back problems?
There are 4 fundamental constraints, starting with the fact that selection can only act on existing variations. It can't generate the perfect trait on demand.
Like the snowshoe hare.
Exactly. As global warming delays snowfall, the hare's molting timing is genetically fixed. It turns white too early, making it highly vulnerable. Selection favors hares that delay molting, but if the necessary allele for a much later change doesn't exist, evolution can't produce it and the population may crash.
Constraint 2. Evolution is limited by historical constraints. It's tinkering, not designing from scratch.
You can't start from a blank slate. Evolution modifies what's already there. A bird has to fly using modified forelimbs. It can't just sprout a third set of perfect wings.
Constraint 3. Adaptations are often trade-offs or compromises.
Optimizing one function often limits another. Look at our highly flexible shoulder joint. It allows for an immense range of motion, but this flexibility comes at the cost of stability, making us highly susceptible to dislocations.
And finally, constraint 4. Chance, selection, and the environment interact.
The environment is constantly changing, and chance events like genetic drift or a random hurricane can limit the degree of perfect adaptation. A population might be perfectly adapted to a given climate, only for that climate to shift unpredictably the next year. Perfection is a moving, unattainable target.
Okay, the accumulation of these microevolutionary changes leads us to macroevolution, the broad patterns above the species level, defined by the process of speciation, where one species splits into 2 or more.
And to talk about speciation, we need a common definition of what a species even is. The most widely used is the biological species concept, or BSC.
This says a species is a group of populations whose members can interbreed in nature and produce viable, fertile offspring.
And they are reproductively isolated from other groups, so the ability to exchange genes is what holds a species together.
But we know the BSC has limitations.
It does. It doesn't apply to fossils or to asexual organisms like bacteria, and it struggles with cases like the growler bear, a hybrid of a polar bear and a grizzly. While they are largely isolated, occasional hybridization proves the capacity to interbreed isn't entirely lost.
So we use other definitions when the BSE fails.
We use the morphological species concept, based on body shape, which is essential for fossils, and the ecological species concept, which defines a species by its ecological niche, which is great for asexual species.
Regardless of the concept, the key to speciation is reproductive isolation barriers that prevent gene flow. Let's break these down.
We can start with the prezygotic barriers, those that block mating or fertilization before a zygote is even formed.
Like habitat isolation.
Two species occupy different habitats in the same area, so they just rarely encounter each other. The apple maggot fly switching from hawthorn trees to apple trees is a great example.
Next, temporal isolation.
They breed at different times, different seasons, or even different times of day. Two skunk species might live in the same area, but one mates in late winter and the other in late summer. No overlap.
And behavioral isolation is huge in animals.
It relies on specific courtship rituals. The unique songs of the eastern and western meadow larks are different enough that they rarely interbreed, even where their ranges overlap. The females just don't recognize the foreign song.
Then you get into the physical blocks, mechanical isolation.
This is when mating is attempted, but morphological differences prevent it. In some snails, the genital openings are arranged such that only opposite spiraled shells can align correctly for mating.
And finally, gametic isolation.
Critical in aquatic species like sea urchins that release gametes into the water. The sperm can't fertilize the egg because species-specific surface proteins on the egg and sperm are incompatible. It's a chemical lock-and-key barrier.
Now, if those prezygotic barriers fail and a hybrid zygote is formed, we get postzygotic barriers.
These prevent the hybrid zygote from developing into a viable, fertile adult. First is reduced hybrid viability. The hybrid's development is incomplete, or it's frail, or it often dies before birth.
Then we have the most famous one, reduced hybrid fertility.
The hybrid is robust, like a mule from a horse and a donkey, but it's sterile. The mule can't produce viable gametes because the horse and donkey chromosomes differ in number and structure, preventing successful meiosis. Gene flow stops right there.
And the final, most complex barrier is hybrid breakdown.
This one's weird. The first-generation hybrids are fertile, but when they mate, their offspring, the second generation, are sterile or frail. The genes from the two parent species just aren't compatible when mixed again.
All right, let's talk about the geography of speciation. The most common route is allopatric speciation.
Meaning other country. This requires a geographic barrier to physically separate populations, cutting off gene flow. This could be a river changeal course, a canyon forming, or the colonization of a remote island. Once isolated, the two gene pools just diverge due to different mutations, selection pressures, and drift.
The snapping shrimp, separated by the Isthmus of Panama, are a perfect natural experiment of this.
They are. When the Isthmus of Panama rose about 3 million years ago, it divided the oceans. Researchers found 15 distinct pairs of snapping shrimp species, one on the Pacific side, one on the Atlantic, that are now fully reproductively isolated. Before the land bridge, they were a single population.
The rare route is sympatric speciation, same country, where speciation happens without geographic isolation. This requires a reproductive barrier to arise quickly within the population.
The most common mechanism for this, particularly in plants, is polyploidy, the accidental production of offspring with extra sets of chromosomes, usually from an error in cell division. If a plant becomes tetraploid, with 4 sets of chromosomes, it can mate with other tetraploids, but it can't produce fertile offspring with the original diploid parent species.
It instantly creates a new reproductively isolated species.
It does. A variation is the allopolyploid, where 2 different species hybridize, and a subsequent error doubles the chromosome number. This new hybrid is instantly isolated from both parent species.
And in animals, sympatric speciation can be driven by strong sexual selection.
The cichlids of Lake Victoria are the classic case. 2 closely related species, one blue and one red, live in the same area. Female mate choice based on male color acts as a prezygotic barrier, keeping the gene pools separate even without physical distance.
So when 2 diverging species meet again, they can form hybrid zones. What are the potential outcomes?
The first is reinforcement. If the hybrids are less fit than either parent, selection strengthens the prezygotic barriers to prevent wasted mating attempts. We see this with European flycatchers. Where the species live in the same area, the males have evolved dramatically different color patterns to reduce hybridization.
The second outcome is fusion.
If the reproductive barriers weaken, the two species may begin to interbreed and fuse back into a single species. This is what's happening with the Lake Victoria cichlids. Pollution made the water murky, so females can't distinguish between blue and red males. This is destroying the behavioral barrier, leading to rampant hybridization.
And the third outcome is stability.
Where hybrids continue to be produced, usually in a narrow, stable area, this persists because individuals from the fitter parent populations keep migrating into the zone.
Finally, what about the tempo of speciation? Is it a slow, gradual march, or does it happen fast?
The fossil record often supports the model of punctuated equilibria. This suggests that species appear suddenly, experience long periods of little change or stasis, and then disappear or change rapidly. The speciation event itself can happen relatively quickly, maybe in thousands of years, which is just a punctuation in a multi-million-year history.
Okay, we've covered the mechanics. Now let's zoom out to the 4.6 billion year history of Earth macroevolution on the grandest, most chaotic scale. And we start at the beginning, the highly speculative origin of life.
The prevailing hypothesis suggests life arose in 4 progressive, unobserved stages. Stage 1, abiotic synthesis of small organic molecules. The early Earth was geologically volatile with an atmosphere rich in water vapor but very little oxygen.
This is the famous Miller-Urey experiment.
Right. They simulated that atmosphere, applied an electrical discharge, and successfully produced amino acids. Later, more accurate simulations of volcanic conditions produced even more. We even have evidence from space. The Murchison meteorite contained over 80 amino acids, confirming their abiotic origin is possible.
Stage 2, macromolecule formation. How did those monomers become polymers?
Researchers showed that dripping solutions of amino acids or RNA nucleotides onto hot sand, clay, or rock causes them to spontaneously polymerize. Heat and a substrate were enough to drive the reaction without enzymes.
Stage 3, protocells. The crucial step of separating the internal chemistry from the outside world.
This involved packaging molecules into membrane-bound droplets or vesicles. Certain lipids can spontaneously self-assemble into these bilayered vesicles in water. These protocells could maintain an internal chemistry different from their surroundings.
And stage 4, self-replicating molecules, the RNA world hypothesis.
Right. RNA, not DNA, was likely the initial genetic material. This is because RNA can do 2 critical things. It can store genetic information, and it can act as a catalyst, an enzyme-like molecule called a ribozyme. Some ribozymes can even catalyze the copying of short RNA strands.
So, early protocells that housed RNA capable of faster, more accurate replication would have been selectively favored.
Natural selection would have acted right there on those protocells, driving the evolution toward more complex, stable information storage, which eventually led to DNA. Once DNA-based life began, the historical timeline becomes much clearer.
So let's review the major milestones. The earliest life forms were single-celled prokaryotes, about 3.5 billion years ago.
And they were Earth's sole inhabitants for 1.5 billion years until the massive global climate shift known as the oxygen revolution.
What caused that?
Photosynthetic prokaryotes, like cyanobacteria, evolved and began pumping O2 into the atmosphere. Initially, this O2 reacted with dissolved iron in the oceans, forming massive banded iron formations. But once the iron was saturated, O2 just flooded the atmosphere.
Which was toxic to most life at the time.
It was. It led to a mass extinction of many anaerobic prokaryotes, but forced the evolution of new metabolic pathways, specifically cellular respiration, which efficiently uses oxygen.
This paved the way for the origin of eukaryotes about 1.8 billion years ago via the serial endosymbiosis hypothesis.
This posits that a larger host cell, likely an archaean, engulfed an aerobic bacterium, which then evolved into the mitochondrion. Later, a separate event involved engulfing a photosynthetic bacterium, which became the plastid or chloroplast.
And the evidence for this is incredibly strong.
Oh, it's undeniable. Both mitochondria and plastids have their own small circular DNA, like bacteria. They replicate independently of the host cell. And critically, their ribosomes are the smaller 7DS type, similar to bacteria, not the 80S ribosomes found in the host cell cytoplasm. They are ancient living relics.
Following eukaryotes, we see multicellularity emerge around 1.3 billion years ago. But the real explosion of forms came with the Cambrian explosion.
Yes, between 535 and 525 million years ago. This marks the sudden appearance of most present-day animal phyla. Before this, large animals were mostly soft-bodied grazers. The Cambrian explosion introduced hard body parts, specialized sensory organs, and large predators with claws, driving this huge evolutionary arms race.
And the last major step was the colonization of land about 500 million years ago.
Moving from water to land required massive adaptations. Plants had to evolve vascular systems to transport water and waxy cuticles to avoid drying out. And interestingly, they seem to have colonized land in partnership with fungi, forming mycorrhizal associations. Animals followed, with the tetrapods evolving from lobe-finned fish around 365 million years ago.
The history of life is not a smooth progression, though. It's shaped by these colossal forces, particularly plate tectonics.
This slow, continuous movement of continents drastically alters habitats and climate. The formation of Pangaea 250 million years ago caused global sea levels to drop, wiping out huge amounts of shallow marine habitat and contributing to mass extinction. Then, when continents break apart, it leads to massive-scale allopatric speciation.
And we have to address the big five mass extinctions.
The Permian mass extinction 252 million years ago was the most devastating, claiming 96% of marine life. It was likely caused by massive volcanic eruptions in Siberia, leading to rapid global warming.
And the Cretaceous mass extinction 66 million years ago is famous for wiping out the dinosaurs.
Right, except for the lineage that led to birds. This event is strongly linked to an asteroid impact. The evidence is a globally distributed thin layer of iridium, an element rare on Earth but common in meteorites dating precisely to 66 million years ago, and the massive Chicxulub crater confirms it.
And a consequence of these catastrophes is the adaptive radiation.
Mass extinctions clear the slate, leaving tons of ecological niches vacant. This leads to bursts of rapid speciation. The most dramatic example is the diversification of mammals. For 100 million years, they were restricted to small, nocturnal roles. After the dinosaurs were gone, mammals underwent a spectacular adaptive radiation, diversifying rapidly to fill those empty roles.
Finally, let's revisit developmental biology. How can slight shifts in developmental genes lead to massive morphological changes?
We talk about heterochrony, which is a change in the rate or timing of developmental events. A minor slowdown in jaw growth in the human lineage resulted in our flattened facial structure and much larger cranium compared to chimpanzees, all from a developmental timing shift.
And then there are the famous Hox genes, the master regulatory genes that control the body plan.
Changes in where these genes are expressed can drastically alter morphology. The difference between a multi-legged crustacean ancestor and a six-legged insect, for example, is linked to a change in the expression of the UBX Hox gene. In insects, UBX acquired the function of actively suppressing late formation in the abdomen.
The ultimate insight here, connecting back to the beginning, is the difference between changing a gene sequence versus changing its regulation.
Changing a gene sequence often affects the protein wherever it's used, which can have bad side effects. But changing gene regulation can limit the change to a specific tissue or developmental stage, making it much easier for selection to favor the trait without unwanted consequences.
And the three-spined stickleback fish beautifully illustrates this.
Marine sticklebacks have large ventral pelvic spines to defend against predators. Many freshwater lake populations where predators are rare and calcium is scarce have lost these spines. Researchers showed this spine loss was not due to a change in the sequence of the PITS1 gene that codes for spine development.
The gene sequence was identical.
But in marine embryos, the PITS1 gene was expressed in the mouth region and the ventral spine region. In the lake embryos, a change occurred in a non-coding enhancer region, a regulatory switch, that turned off the gene's expression specifically in the spine area, but left it active in the mouth where it's still needed.
It's definitive proof that altering the switch is a fast, efficient path for evolutionary change.
It is.
So to conclude this incredible history, we have to reiterate that evolution is not goal-oriented.
It's a historical process of continuous modification based on immediate survival. Structures that evolve in one context are frequently co-opted for a new function. Again, our jaw hinge bones becoming ear bones were not designed to be auditory. They were repurposed structures that just happened to be useful.
And the idea of an evolutionary trend, like the horse evolving toward a single toe, is misleading if you only look at one branch.
The horse family fossil record is a massively branched bush with multiple lineages going extinct. The current genus, Equus, is just one surviving twig. Trends like increasing size are simply the result of strong directional selection in a specific environment, open grasslands, not some intrinsic drive toward a predetermined perfect form.
We have journeyed through 4 billion years of life, from the simple logic of Malthus to the grand chaos of plate tectonics and asteroid impacts. We've seen that evolution is a historical process guided by natural selection, influenced by chance, where speciation arises from reproductive barriers, and macroevolutionary patterns are dictated by these massive geological and catastrophic forces.
And let us end where we left off with the cichlids in Lake Victoria. We noted that human-induced pollution caused the water to become murky, breaking down the female's ability to use color for mate choice. This fragile million-year-old mechanism of reproductive isolation was destroyed by environmental instability, leading to fusion and species loss.
That reveals a profound fragility. If human activities, from pollution to rapid climate change, are actively accelerating environmental instability, what does that mean for the future of life on Earth?
It raises the ultimate provocative question. Given the constraints on evolution, that it only tinkers with what it has, and it can't create perfect solutions on demand, which forms of life, whether highly adaptable pathogens or organisms with immense reproductive capacity, are best suited to thrive in our increasingly unstable global environment, and which ancient, specialized lineages are we putting irrevocably at risk?
A truly profound thought to mull over. Thank you for joining us for this deep dive into the mechanisms of life.